Well, hello everybody. I'm Steve.
And I'm Dennis.
And we'd like to thank you to another Natural Growing Through Biology. Biology. We're going to be talking about something a little bit different today. We're going to be talking about pies.
We're talking
Strawberry
about
rhubarb?
biology, phosphorus solubilization with biology. That's
like
why
that.
you said biology.
I like that, but I like strawberry rhubarb, so we can talk about that. So, biology podcast series, and we are going to be talking about nutrition and nutrients today. We're going to be talking about biofertilization and how microbes are absolutely critical, just essential for the uptake of the nutrients, and specifically some. In this case, we're going to talk about phosphorus because it is so difficult to think about getting phosphorus into a plant efficiently without utilizing the microbes in the soil to make that reaction occur.
And this is a common question that we get all the time across the board in all crops. This isn't just specific to a few plants. I mean, this is across the board in all different soils, all different agricultural practices.
Globally, we talk to people about this literally every day. Yeah. Every day, everywhere. Phosphorus, my plant's having trouble getting phosphorus up. How do I get phosphorus up? So we're going to talk a little bit about that. And we're going to start off and look a little bit about, the history, this idea of what has been taught, what is so ingrained in what people think about when they think about nutrition, they think about fertility, and they think about fertilizing plants. It's this old mentality of me dump fertilizer in water, plant goes like a straw, sucks it up, and that's it. That's the end of it, right?
And the amazing part about that is, even when you Google this and you utilize AI and this artificial intelligence and all of this stuff, I Googled it just to see.
I know, I like how you looked that up. It's fun.
I know, and it still says basically the same thing.
Yep.
And does that happen? Yes. That's all part of nutrient availability in some way.
Mass flow happens.
Exactly.
Osmosis happens, yes.
But what we're going to talk a little bit today, more focus us on strictly what we feel is the most efficient way to utilize some of these things through soil digestion and biological digestion of making this nutrient available.
Well, this is kind of like people talked about leaky gut for years. This is the idea of a leaky gut where the nutrition is just literally flowing in versus the microbes in your gut digesting, processing, and making that food into a bioavailable form. And just like in our gut, if we have a leaky gut and that nutrition is leaking across and going past the microbe barrier, we have health issues. It is exactly the same as with the plant. When we see some of those deficiencies occur and we see some of that system degraded, we're naturally going to be fighting harder and harder. So we have to focus on how do we fix that system?
Well, yeah, and and the same thing on the AI, when I looked at that, it still talks about, organic decomposition. It talks about nitrogen fixation, but only talks about nitrogen fixation with rhizobia.
Yes,
it doesn't talk about free living nitrogen fixation in all crops or endophytic nitrogen fixation. It talks about mycorrhizae and phosphorus availability, but we have gone through the research now that we know it has went well beyond that. And one of the things we talk about, is Dr. White's research.
Absolutely critical. When we start thinking about how do plants get nutrition, we need to get past this idea of root hairs or straws that are sucking the nutrients up. That's not what's happening. What we found is root hairs are actually a way for the plant to deposit more of those microbes that it wants and needs into the soil environment so that they can go out and mine and harvest and extract. We need to think about, you're a farmer, your plant is also a farmer. It is purposely growing a crop of microbes so that it can go out and extract the food. Just like we're extracting food from our crops, the plant is extracting food from its microbe crop.
Yeah, and it does that within that soil environment. And we're not going to talk a whole lot about this. We did this last week, I think, in reduced inputs and in some of our other conversations. Last month, I guess, our last podcast, whatever we did. It was yesterday to me.
Sure.
But anyway, when we talk about how those microbes and those exudates actually manage that, and we'll talk a little bit about that later today, but it's pretty amazing when you start to think about this rhizophagy cycle.
Or rhizophagy.
And exactly what goes on within that soil environment. It's pretty amazing.
Yeah, so we need to think about this rhizophagy, this plant farming microbe, plant eating microbe, and extracting nutrition from the microbe. That's how plants have been feeding themselves forever, for a very, very, very long time. Plants have been feeding themselves without human interaction for millions of years. it's our emphasis on we can do it better. I know that we can do it better. We built these tools. The war machine basically created this phosphochemical approach where we're utilizing all of these different inputs, all these different chemicals, all these pesticides to combat these issues that naturally happen in nature. But nature naturally has solutions for them. And when we look at a healthy system that's been extracting nutrition through microbes, we see a much more balanced nutritional profile. And that's because microbes, just like the plant, just like any other living organism, they don't need just NPK. They need the zinc. They need the manganese. They need the copper. They need the iodine. They need selenium. They need all of these other nutrients so that they can be fully functional and fully formed and produce their own enzymes and their own microbial communication molecules. So they are a living system that is utilizing all this nutrition from the soil around them to live. And a plant, by extracting and consuming those, is going to have access to nutrition. And we'll talk a little bit here at the end, looking at the difference when we're utilizing a more conventional versus a more biological. And it makes sense. We see significant differences across the board in nutrition when we take Mother Nature's digestive system out of the equation. And we need to remember that all of this microbial, all of the soil economy, all of life runs on carbon. So we start with carbon, we end with carbon, and carbon everywhere in between. So when we're talking to growers, and we are always saying a couple things, test, don't guess. We've got to evaluate to know where we are so we can know where to go. And we also talk a lot about it's a program, not a product. And this idea of carbon infusion, soluble carbon, utilizing organic inputs, fish, humate, seaweed, all of these things are a way for us to get these microbes through a struggle time when we put them out in the soil before that plant is really photosynthesizing and creating exudates to feed the microbe. Because you're always talking about that's the best, yeah?
Oh yeah, obviously the best food for a microbe is the root exudates, so those communication molecules that are coming from that plant. That is by far the best food. Yeah. But one of the things, I always refer to this, put them out with a sack lunch, and not only does it feed them, but it also protects them. So when we put them into a tank mix and we're mixing it with maybe some of our other things, to throw a little bit of that carbon in, which is a buffering, it protects that biology and it puts them out into that soil environment so that when they wake up and they come out of dormancy, they immediately have food. They have something to get energy to start that process, because that's really what we're looking at. Is we're taking these guys from an insistent dormant state, we're inoculating them into a whole new environment. Let's send them out with a little bit of a care package.
Yeah, we don't want hungry, angry bears.
Oh,
we
don't
want well-fed, happy bears.
Exactly.
So that is kind of a great transition, and talking about this idea of extracting nutrients, utilizing microbes, and how these microbes also require high levels of phosphorus. When we look at a microbe, its profile, it has ATP just like a plant, adenosine triphosphate. Its phospholipid bilayer, every membrane that surrounds every single microbe, every single cell, is made up of phosphorus and fat. And fat is one of the highest energy storage molecules known to man. So as a plant breaks these materials down, it's extracting all this nutrition from the microbes. And we'll talk a little bit more about there are specific microbes that do a better job of it. When we look at the microbe and think about it kind of as a supplement, most microbe, the basic profile, is a 10-2-2 plus everything else. If we get into a nitrogen fixer, instead of a 10, it might be a 15, might be an 18. When we get into a phosphate-solubilizer, all of a sudden, instead of a 2, we can see 4, 5, 8. We can see an uptick of that. So Dennis talked about this idea of a plant communicating its need. The beauty is it can communicate specific nutrient needs as well. It's going to feed and attract specific microbes that have the capacity that it needs. It's going to then take them in through its apical meristem on that root tip that's growing, and it's going, we still don't fully understand how, but it's able to identify the microbes that it wants to replicate, and it literally purposely clones the ones that it wants. The ones that, if it needs phosphorus, it's going to clone the organisms that have a higher phosphorus level so that it has naturally more phosphorus uptake. It then utilizes those root hairs, spits them out, and now we have, instead of 5 or 6 phosphate solubilizers, we have 5 or 6 million phosphate solubilizers, allowing that plant to extract naturally what's in the soil.
Well, and that's some of the idea that comes back from the research that's being out there now based off of foliar nutrition, where you use a very small amount of a nutrient, you put a ply of foliar and you think, how can 8 ounces of this or 4 ounces of that make a difference in overall plant health per acre? But what they're finding, and the research is actually showing, is that is actually stimulating that plant.
Yes.
It uptakes that, but then it wants more. And then that communication goes down into that soil, which makes more of that available. And I mean, it's pretty amazing when we start to think about how complex this system is in agriculture that we sometimes take for granted that we have total control over.
Yep.
But if you really think about it, if we I just had this conversation today and talking with some growers overseas, if what we do as a grower sometimes is the greatest reducing factor to overall plant health, because we do not understand the actual function of that biological system and that plant need. We interrupt that with excesses, which then create imbalances within that plant.
It compounds, it snowballs. No, as soon as we try to put control on the system, we're now responsible for that system. So any of the errors, any of the mistakes that happen, as we reduce that biology functioning as a buffer on and around that plant, we are now responsible. And generally the end outcome is the Isle of Death. We end up out there trying to control something. We try to control a nutrient, we try to control the plant, and now we end up trying to control the pests that arise. So as we're talking about phosphorus, we're talking about soil and nutrient extraction, we need to look at what's going on. We need to look to the future because, we're farming this year, we're farming next year, but a lot of farmers out there, these are multi-generational farms. So we need to have a long view perspective on what is going on in the world, what's going on in the soil, what's going on in our oceans as we see massive dead zones, anoxic zones because of the phosphorus that's leaching off our farms. We know that we're losing phosphorus. And if we look globally, where are the phosphorus reserves? We see pretty quickly that most of the phosphorus, 75% approximately, are in mines in northern Africa. That's where most of the phosphorus in the world is. And looking at research, there's some discrepancies. Some research is saying slightly longer, some research is saying slightly shorter. But a lot of research is starting to say we're about to hit peak phosphorus in the next 10, 15 years. And from there, we're on a downward decline. We're going to start being able to extract less. It becomes more difficult to extract, so phosphorus prices are going to go up. We need to be more efficient. And a lot of research is saying by around 2100, we're going to be declined to the point where it becomes a very limiting resource, and we're going to start to really have to pay close attention to what that phosphorus is, where we get that phosphorus, and how to stabilize that phosphorus. And one of the beautiful things about living systems is they are naturally designed to accumulate and utilize phosphorus. Phosphorus, like we talked about, functions is ATP, the energy transport of the plant. It stimulates early growth in the plant, stimulates root growth. So as we're trying to mine and extract more nutrition from the soil, that takes a lot of energy. It takes a lot of exudates, takes a lot of nutrition, and that plant is able to purposefully grow its roots in directions that have a higher level of nutrient it requires. And a lot of that has to do with back-and-forth communication. Microbes are on a smaller scale than the root, even the root hair, even the mycorrhizal fungi, mycelium that are down in the soil. So they have access to these zones, and they're spitting out communication molecules, and they can kind of communicate and tell the mycorrhizae and tell the plant where some of these pockets of water are, where some of these pockets of nitrogen, organic matter, phosphorus, where these are naturally in the soil, to allow it to have better access. We see a much better seed production, much better germination of the seed. Phosphorus is a critical component of DNA. It's a critical component of all the structures, like we talked about, that phospholipid bilayer. So we start to realize that not only is phosphorus, we've talked about in the beginning, it's difficult to extract from the soil. We'll talk a little bit more about that. We're going to have a very limited resource of it, and we've got to have very close reins on it. We've got to really know what's going on with that. And that's why there are a number of different phosphorus analysis techniques so that we can see what's going on in our soil. Because this idea of luxury nutrition is going to provide us the highest levels of yield. We were just listening to David Knaus.
Yeah.
A great presentation. He's talking about this idea of luxury. We've got to get over it, because that luxury level it's just going to shove something out of balance, just like you were saying.
Yeah, we talk all the time about how important balance is and how excesses create a much greater problem than a deficiency. We've been talking about that for 30 years. And you're starting to hear more and more of that understanding, especially with leaf extract analysis or plant sap. And that's what David was referring to the other day in the presentation.
You can't hide it. It's right there.
This luxury is actually creating problems with something else that we can actually struggle with. And so, we want sufficient levels or maybe a little bit, but when we get to those excesses, it just plays such a role in overall plant health and nutrient root development, nutrient density. All of these things we're talking about in regenerative agriculture, biological agriculture, we refer to it as farming. I mean, it's farming, is related to nutritional balance and microbial diversity within that soil environment, microbial populations, soil structure, soil health. All of these are really direct correlation of plant health. They help with plant health, but they also can reduce soil health if we don't have plant health. I talk so much about the diversity and the function of beneficial organisms within that soil environment, create a living root, a living plant that is actually photosynthesizing. And we start to stress the plant, it starts to reduce those exudates within that soil environment. And that's why we talk about phosphorus. And phosphorus is a key role in overall plant health.
It's one of those NPKs. It's right there in the NPK.
Exactly. But we need it from the establishment, from germination, all the way through seed development and harvest. It's critical. It's a critical component through those entire processes.
It's always needed during certain phases of the plant's growth. It's needed at even higher quantities. And sometimes, if we think about what's happening when most of our plants are really setting seed and putting their all of their energy into the basket of the next generation, well, it's usually fairly hot. It's usually fairly dry. Our soils are drying out. And if we haven't done the things to build our aggregate levels, to build that organic matter, to utilize those exudates the plant is releasing, to build soil, it compounds. And without those microbes having access to that, and without the plant having access to the microbes, its ability to extract phosphorus goes down considerably. And then we rely more and more on additions and applications, which can push things even further out of balance.
But the thing about that is we talk about it, and you talk about it all the time, Steve, when we apply this phosphorus to that soil environment, all of the environmental impact that it has based off the availability of that phosphorus. I mean, when you start to think about it, we already said, you talked about the reserves going down. We talk about, okay, now we're applying excess phosphorus. And so much of that, when we think of it without biological activity, the availability of that phosphorus is very limited.
Oh, absolutely. So in an ideal environment, with the ideal pH, with the ideal temperature, ideal moisture, all of these, if we apply $100 worth of phosphorus, at the maximum, $25 becomes available to the plant under normal fertilization conventional practices. It's just not available. If our pH is relatively high, tricalcium phosphates form. If our pH is relatively low, iron and aluminum phosphates form. If the temperature is low, we see a significant reduction. At 70 degrees soil temperature, 70 degrees Fahrenheit, soil temperature, we see roughly, that's about our max. We see about 100% availability. As we drop down to 60, we're less than 50% availability. It is that critical. And that's why when we talk about the importance of establishing these microbes early in the spring to feed those plants, that's a big part of it. We've got to start that system early. And that's why we're very, very big fans of in-furrow at time of planting, seed inoculums, anything we can do to get those beneficial organisms at a high level on and around the plant is critical. Because we've got to look at the soil as an overall profile, don't we?
Yeah, and so often when we talk about that phosphorus availability and that temperature of 50 degrees and the reduced availability of phosphorus, a lot of times that's at the point of seed germination in a lot of our crops. And that's when that plant actually needs that phosphorus. So we apply that phosphorus. You talk about only $25 out of $100 available. So what happens to the rest of that phosphorus? Where is it? This is kind of the idea that I love when we start to look at this total nutrient digest. When we've switched from a soil analysis to a total nutrient digest, to where we start to see what is actually in that soil environment. And this is an example from Ward Labs, which talks about the phosphorus levels 878 parts per million, or pounds per acre available within that soil environment. So there is plenty of phosphorus there. It's just, as you talked about, so little of that is really available. And when we start to talk about the availability of it, plus the temperature relationship of the availability of it, plus the tie-up of the availability, all of these things start to look at and lead us to the world of biology. When you start to talk about what does phosphorus bind with? The phosphorus, we go back to the calcium, we go back to the iron, we go all these things that it binds to. The only way to release that is through microbial activity and enzymes.
Absolutely. So phosphate-solubilizing bacteria, PSB, or kind of what we constantly refer to them as, have the capacity to extract these nutrients. And they're utilizing different organic acids, they're utilizing different enzymes. So if it's a low pH, they're going to use acid phosphatases to extract it from the iron, extract it from the aluminum, which can also help with some iron uptake. The other side of that is if our pH is slightly higher, they're going to use alkaline phosphatases and tricalcium phosphatases to split that phosphorus off of the molecule to make it available. And then we also see calcium uptake go up. So when we're looking at our soil environment, we're looking at our pH, we're looking at all of these different factors. As we always say, test, don't guess. All of these are pieces of the puzzle to give us an indication of what's going to happen and how that system is going to perform. And the beauty is some of these phosphate-solubilizing bacteria is really cool. They form what's known as a polyphosphate cyst, which is basically their own storage. So inside of these little supplement pills, the microbes are creating their own little micro supplement pills. They're purposefully hyperaccumulating little tiny beads of phosphorus so that when times are lean, when temperatures aren't quite right, when moisture isn't quite right, when they have difficulty accessing the phosphorus, they have access so that they can continue to reproduce. And remember, through rhizophagy, all those little beads, as the plant makes those microbes swell up and leak or swell up and burst, all of that is now available to the plant. And that's where we see a massive shift in this idea of water-soluble phosphorus applications to this idea of biological phosphorus applications, where we're changing the system to allow the microbes to do the extraction.
And how often have I talked with a grower where we do a normal acid extract analysis and we look and let's say our P1 levels are below 30. They're down around 18 parts per million. And we have a very high pH and the grower feels, well, it's not going to be available. I need to put out some phosphorus. But then when we use these phosphate-solubilizing bacteria and we use these biological practices, suddenly we see from a plant sap or a leaf extract,
no, you're fine,
that the plant has all the phosphorus it needs. It's in a sufficient level. And if you looked at a soil analysis, well, that goes back to the idea of this total nutrient digest. It's there. It doesn't show up on a normal acid extract test, but it's there in that environment. And that's what these microbes are utilizing. I mean, they're utilizing that to create these phosphate prills inside their body because that's what they need in order to survive.
Yep. As the plant calls for phosphorus, it increases the number of those phosphate-solubilizing bacteria. Their phospholipid bilayers are chock full of phosphorus and energy. They're creating these little tiny prills, little fertilizer beads. They're creating their little micro fertilizer beads, which is just fantastic. There's also polyhydroxyalkonates. There's microbes that will hyperaccumulate their own carbon in different ways so that, again, if times get lean, they can continue to grow. But we're going to go past that. And we were at a presentation here fairly recently put on by the Spokane Conservation District.
Yep.
So cool. They had Joel Williams over, and we stole. I mean, he gave us permission, but we straight up stole some of his slides just because It's stuff that we've been talking about for a long time, but when somebody else says it, sometimes it feels good. It feels good to have somebody else say the same things that you've been talking about, because we're not, I mean, we're crazy, but we're not that crazy, right?
Yeah. Well, we're pretty crazy.
Okay.
Yeah. But, we used to talk, I used to talk about the grocery warehouse and the idea of availability and nutrition. And Joel kind of put this out there, the two schools of thought of how do we build this biological community within that soil environment for plant health. And really, all of these principles have to be applied in order to build soil health. But he talked about stimulating or feeding the native organisms through soil health, through plant diversity, through feeding that soil, feeding that biological food.
Biostimulants.
And then, and then introducing or inoculating based off of soil applied, or these organisms basically within that soil, or plant applied seed coatings, all of these things that we talk about, and how we build that biological diversity within that soil environment. And a lot of times, you always give the example, Steve, of biological inoculums are no different than a cover crop seed. We're seeding that environment with these organisms in order to help that environment grow, to create those structures, to create those environments. A lot of times what we look at it as these are, as we say, plant growth promoting rhizobacteria. They're designed to enhance plant growth. Phosphorus availability, nitrogen fixation, potassium mobilization, calcium, all of these things, trace mineral profiles, they're designed to make these more available, readily available to the plant, so that the plant can actually use them, so it can photosynthesize better, so we get all of these crop health ideas. I mean, that's really what it comes down to.
The microbes, they're like every other living organism. They're selfish. Their goal is to reproduce. If they can create an environment that the plant is thriving in and photosynthesizing more, you know what happened? More exudates. More exudates, more food for microbes, more babies for the microbes. It is a self-serving system, but it happens to be a symbiotic system, allowing both of these systems to function better. Another side of this idea of introducing or utilizing natives is the idea of, are we going to focus on our plants, or are we going to focus on the bulk soil? Focusing on the bulk soil, there are a few approaches that we utilize that kind of make a lot more sense for that than trying to focus on specific seed zone. When we're working with a grower that has a lot of field debris, a lot of stubble, they've had a spill, they've done something to a large area, we've got to look at a bulk soil application. We've got to look at a larger area application to create that digestion. It's usually a digestion that we're trying to do, a decomposition, whether it's chemical, whether it's organic matter. We're trying to work on a large area. Most of the time when we're talking about plant growth promoting rhizobacteria, like our Spectrum product, we are trying to put it on the seed, because that's where we're going to see the benefit. We might not have a lot of benefit. We might not see a lot of those microbes out in the bulk soil. So if you're doing genetic analysis and you're trying to analyze the bulk soil and, you said I put out these phosphate-solubilizers and I'm not seeing them out here. Well, you didn't look for them where they're going to be. They're going to be on and around that plant. These are rhizobacteria, and they are creating what's known as a rhizosheath. They're creating this buffer around the plant root that is a completely different environment. They can adjust the pH, they can adjust the oxygen levels, they can adjust how that system is going to function. They're going to increase the organic matter considerably, which allows better gas exchange, which allows better moisture holding capacity, allows better nutrient extraction. Again, all of this is to create an environment for the plant to thrive.
Well, and we talk about that all the time as the idea, it's not, these inoculums aren't trying to change the bulk soil. They're not trying to change the makeup of the organisms within that soil environment. I mean, we had some testing from Montana State that basically talked about that we did a 4-year study on, just by applying beneficial bacteria, we saw an increase in fungal, not only fungal growth, but fungal diversity within that soil environment. So it was kind of significant it goes back to the idea of, if we're around that root and that plant is functioning at a higher level, it creates habitat for everybody else to come in and live within that soil environment. And that's really kind of what we're trying to do here.
If you build it,
they will come. I I look back, that presentation I did, the first time I did that was in 1999.
Wow, yeah. That's awesome.
Yeah, it was a slide deck with an overhead projector, though. It was a little different world back then.
How did you have your animations work of like you going across?
I would, yeah.
You just like moved it with a popsicle stick, a little popsicle stick?
Yeah.
Love it.
But no, I mean, and it's actually funny as we talk about that rhizophagy or that zone around those microbes, and we start to talk about that pH, and it's pretty amazing. I know this is a podcast, but there'll be some pictures in along with this that Dylan will make sure are available. Yeah. But this was a potato plant, which was pretty amazing when we went out there and dug this up in the field. Is this plant is not 4 inches tall, and yet the root mass on the potato plant was just incredibly unbelievable, and you can start to see the hooking that is starting there around that. But this is what we're talking about when we create that in soil environment. Those roots are there to feed that plant. They're creating that biological world, and it's really pretty amazing when you start to talk about, we've talked a lot about the pH of how this rhizosheath creates that pH around that plant for the environment that the plant wants to live. But you see, it does a lot of that based off a high pH or low pH.
Yes, depending on what is needed, these microbes and the plant communicate together and they create the environment that is going to work the best for the plant at that given time. If the plant needs more phosphorus, it's going to shift and change. If it needs more nitrogen, it's going to shift and change. It has the capacity to change this environment directly around it in that rhizosheath, in that rhizozone. So we look at this gigantic root ball of potato and we say, well, that's a large area. And we start to, if you zoom in even closer, you start to see there are little micro aggregates that are holding all of this together. Those micro aggregates are then bound together by bacteria with their extracellular polysaccharides or extracellular polymeric substances, because we're starting to say it's not just sugar. It's enzymes, it's DNA, it's all these other things that they're spitting out in there. But anywho, sorry, I digress. So these microbes are starting to create those little individual clay particles, sand particles. They're starting to stick them together. And then we take a few of those, and you have a fungal community, like you were just talking about, increasing the alpha diversity of those fungal species. Then we start to see larger macro aggregates form. So we start to literally physically build soil from the plant's root out.
Well, and that's what, we talk about basically not changing the bulk soil of the biological diversity.
That's hard. It's like trying to change your pH.
Exactly it. Trying to change your pH or trying to basically, as I say, in some cases, all of the implements in the world based off of whether it's calcium, based off of whether it's potassium, based off of whether it's phosphorus. We apply tons of these products and we pull a soil sample and the soil sample says the same thing. We're not making this change. No. I mean, that's kind of what I look at in this biological. We're changing it around the plant. And we do the same thing with the pH. We've talked about this a lot. People have used sulfur or gypsum or whatever the scenario might be because my pH is a 8, my pH is a 4. A lot of times in the biological world, they're creating an environment to where they want to live, and they're shifting that environment based off of communication with the plant, which is best for that plant to grow, kind of like what you're saying. I mean, it's not the bulk soil. Nope. We're not changing the pH anywhere there. Nope. We're just changing it around that actively growing root.
Well, my blueberries, they have to have a low pH because they have to have an ammoniacal, blah, blah, blah. Well, no. If we are going with conventional fertility, conventional mindset, I agree. That's the only way you're going to maintain that system. Or we get out of the way and let the microbes do it. And we start to see that the entire system shifts and changes. This is looking at this picture, which again, sorry podcast people, but Dylan's got your back. We're looking at an inoculated plant versus an uninoculated plant and looking at the exudate profiles that we see associated with it. With this uninoculated plant, there aren't exudates. The plant shuts them down. When it's in a sterile environment, there is no need, there is no reason for it to do that. So we see less carbon, less exudate going out in the root environment, which is going to then attract fewer microbes. It's going to be out there as a naked bare root, which is exposed to any and everything. Temperature change, moisture change, pH change. Without that rhizosheath to protect it, that root is exposed to all of it. Versus inoculating, we start to see this plant pumps out massive amounts of exudates, and that is purposeful as communication molecules. That's what these exudates are. It's creating a zone right around the root that encourages specific reactions to occur. And some of that is a redox. Some of these nutrients that we need have to be in a specific redox state. You have to have your manganese at a specific charge. You have to have your iron at specific charges. Those microbes are going to create the environment that shifts and changes that. I think back there years ago, there was some work done in the Hanford Reach area, and they were using molasses to feed Bacillus megaterium for the hexavalent chromium, and they saw that the Bacillus megaterium was changing that hexavalent to a less carcinogenic, a less damaging form naturally on its own. So we see these microbes doing things that we don't really expect them to do. They're able to bind up salts. They're able to bind up heavy metals. They're able to take this environment and completely shift and change it. Again, they want a happy, healthy plant.
Well, and looking at those root exudate, it kind of reminds me, for those people that are on a podcast, of having a perfect white tablecloth on a picnic and you lay a root on it, but then having a perfect white tablecloth at a picnic and you dump red Kool-Aid on it.
Or strawberry rhubarb pie.
Or strawberry rhubarb pie. You've stained it. And I mean, that's really, for your visual, I mean, it's that's that what we see. Visual as what we see for those exudate profiles versus a plant that has been inoculated or has those beneficial organisms, or a plant that does not.
Yep.
I mean,
night and day.
Night and day.
Yeah. And we physically see the plant turning on its exudates and turning off its exudates. And this is research. This is in a lab. This is extreme scenario. But we get the point that these microbes are necessary. The plant has to sense them in order for this communication, this system to function. So when we go out and we completely attempt to sterilize the soil, we have major ramifications, major impacts that are going to occur. Because without those microbes, the plant is exposed to, like we talked about, temperature shifts, UV radiation. Some of these microbes in the soil communicate with the plant and help upregulate and help feed the plant UV protective molecules. They're also doing the same with insecticidal or pathogenic organisms. These microbes talk to the plant and they allow it to increase its own defensive molecule capacity, whether that's in the soil or it's in the phylosphere. And that's kind of the cool thing. This graphic is showing the idea of utilizing, this is a foliar spray. Bacillus subtilis and Azospirillum braziliensis are both considered rhizobacteria. In this research, they're using these quote-unquote rhizobacteria as a foliar spray. And in doing so, they see the same shifts and same upregulation of the plant secondary metabolites, the protective molecules. They see upregulation of amino acid, or therefore protein synthesis, lipopolysaccharide production. They see all of these same shifts that you'd expect from the soil microbes happening on the leaf tissue. And we can even see better phosphate uptake both on the leaf and in the root when we start utilizing some of these microbes as a foliar spray. So I know we roots, rhizo, but we need to remember that the leaves are there too, and they are doing an incredible thing. And we have to think about them the same way. We're always talking about, feeding our roots, protecting our roots, making our roots into an environment that we don't have to worry about stresses. We can do the same thing with our leaves. Well, we can treat that plant in a way that allows it to protect itself and change itself.
Well, and I talked early on in this presentation about phosphorus uptake and the idea of foliar applications of phosphorus and increasing basically the uptake through the soil.
Yeah.
And I see that all the time with these. Everything that happens in the rhizosphere also happens on the phylosphere with nutrient uptake, with those microbial communities. All of the exact same things happen. We just have a tendency not to talk about it as much. But I mean, when you start to talk about this, by doing a phosphate-solubilizing bacteria on the phylosphere of the plant, increases phosphorus uptake.
Yep. So, this isn't a one-way road. It's not from root to leaf. We've talked about the exudates. It's from leaf to root as well. And it's not just sugar, it's communication molecules. It's different enzymes, different acids, it's different plant secondary metabolites. It's all of this milieu. It's this milkshake of yum that the microbes want.
A milkshake of yum?
Milkshake of yum. That's what plants are doing all day, every day.
Well, and is there any research out there that kind of proves what these two crazy nuts are talking about?
That calls it a milkshake of yum? No, I think that's
yours?
That's a Steve exclusive right now. But no, look, and I pulled up a few. This is looking specifically at organisms associated with phosphate solubilization in different plants. And there are sheets and sheets and reams and reams. This is not new research. This is not new to science. In some cases, it's new to some of the agricultural communities. It's not new to science. This has been going on forever. It's just we've got to get past some of that mentality of feed plants what I want, NPK, salt-based to get it to grow and maximum and blah.
Well, and that's part of what we talked about when you searched at the AI that I did in the very beginning, which talks about how plants uptake is, this regenerative agriculture, the growers are starting to understand that there is a better way to do this.
Yep.
And managing how we do this with these microbial communities and feeding the diversity of these microbial communities, however we decide to choose to do that. But so much of that comes down to, and today this presentation is about phosphorus solubilization and making more phosphorus available to your plant. But I mean, we have some research of our own that we have done out there that we kind of started when we developed this product. And it's kind of interesting because for the phosphorus, the cannabis guys, I learned more about cannabis when it was legalized in Colorado, whenever that was years ago,
right?
Than I ever thought I would know about that crop. But the cool part about that is these guys pay great attention to detail.
Tightly regulated, tightly controlled, tightly monitored.
And inside a growing environment in a laboratory, when they have a problem, they can come to you and say, I need more phosphorus. What can I do? So you create a protocol for them, and we build like the Spectrum PSB, and you say, utilize some of this product. And what was unique was one of the cannabis growers basically inoculated a control and a treated.
Clones, exact same environment.
Exact same environment, same clones, same growing medium, same nutrition package, and they did it with a cover crop, which is kind of interesting in the cannabis world.
I love that.
Yeah, and so they did that, and what they saw is earlier germination,
about 2 weeks
earlier, of germination of not only of all of the cover crop seed that they had put down there, but they also saw an expression which was significantly different in the growth rate of the cannabis plant from the two different applications. And the really interesting part is by the time the control had germinated the cover crop and was starting to grow, the other cannabis plants were on their way and actually branching and producing.
And about 2 weeks.
2 weeks. The only thing different was phosphate-solubilizing bacteria, making that nutrition available to that for all of those processes to happen.
And we talk about the importance of plant secondary metabolites. A lot of those essential oils, that's the flavor. That's what you want out of your crop. That's what people are eating and tasting and consuming. But remember, oils, like I mentioned, the phospholipid bilayer, lipids are one of the highest energy storage molecules. That means it takes a lot of energy to build them. Our plant secondary metabolites, essential oils, are these lipid-like and lipid-based molecules. It takes a lot of energy. And if we think about phosphorus as the little microscopic battery that allows these systems to function, that is our energy storage, allowing this transition to occur. As we increase phosphorus, as we allow this plant to have better access and a more balanced access, it's able to produce more of these protective molecules, essential oil molecules, flavor molecules, all of that. Well, all of that goes up.
We see some of that in essential oil production. You know, your lavender, your tans, any of those, Melissa, all of those, the same idea.
Yeah, we worked with those growers as well, and it's really exciting to see the oil contents go up.
Yeah, and the oil quality.
And quality, yeah.
Yeah.
This one was a few years back. This was, again, some of the initial, it's hard to believe it's been over a decade.
Yeah.
Since we started doing some of this focused research. This is a grass grower. Very purple, very obvious phosphorus deficiency in the crop. They went out, they applied phosphate-solubilizing bacteria, in this case Spectrum PSB. And the cool thing is you can see the line of where that plant now had access to adequate phosphorus. What's already grown, that's done. That's already in the past. The plant generally isn't going to do a lot about that. Its new growth is now green and healthy. And so we see this a lot of times when we're working with crops and they've got a tip burn or they've got damage or they've got a mobile nutrient issue on the lower leaves. Those lower leaves, they're gone. Yeah. It uses them as sacrificial, but we can grow out a lot of those stress environments, whether it's sodium, whether it's phosphorus, whether it's nitrogen. All of these things can be overcome with microbes and nutrition.
You know, and that gives me an example of a corn grower that we had in the Othello area. Was there's half a field that was grown under normal practice and half under the biological, and both sides were showing a phosphorus deficiency early on in the corn. And PSB was applied to half of the field.
Yep.
And it's interesting because you can say, well, the temperature changed or it became available or whatever. The field that was not treated with the phosphate-solubilizing bacteria continued the deficiency, where the side that was treated with it now grew out of it. And the interesting part about that, it was so visual to that grower, he treated the control side with the phosphate solubilizing to catch it up because he was worried about, this was for silage, he was worried about his yield.
Oh, we lost our data.
that is so cool.
Yeah.
But it was cool that he saw that.
It works.
Yeah.
And we need, so we've talked a little bit about grasses, we've talked a little bit about cannabis, but we can also talk about nitrogen fixing crops. Nitrogen fixation is the most energetically expensive process that happens biologically that we know of. It takes a massive amount of energy for nitrogen fixation to occur. So when we look at our legumes, when we look at our rhizobia crops, when we look at our crops that have nodules that should be fixing nitrogen, it takes energy. And whenever we talk about it takes a lot of energy to do, phosphorus is going to be one of the ways that we want to make sure we have ample levels of phosphorus to feed these highly energetic processes that occur in nature. So in this case, we're out there, were these chickpeas or garbanzos?
No, I think they were
chickbonzos. Chickbonzos. Okay, these were chickbonzos. And we can see the root development significantly greater. We need to have a biomass reading on this. We need to bring our scale out to the field more often. But we can see visually massive growth difference. Harder to see in the photos, but the nodule size, occurrence, frequency, we saw a lot more nodules that were a nice dark pink, and they were much larger on these treated
And
plants.
I got excited about the nodules, where the grower got excited about the root mass, because the availability to carry those through a drought period with that better root structure. And the other thing that we noticed is just, especially if you look at the picture on the left, the stem size. Not only the overall plant health, but the stem size of that plant. How much healthier that was.
That's a 2-lane highway versus a 4-lane highway. It's able to move a lot more energy,
material,
more nutrition. It's also a much more resilient, it's a stronger structure if we build it right. With silica, with calcium, with all of those other things that we can get from the microbes. And then we started looking at pod counts.
Yep, we looked at pod counts there. You know, it was basically the treated standard program of growing the garbs was 12 pods. You know, on the T1 treated, which was a different biological program, but the idea behind phosphate-solubilizing bacteria was 24 pods. And then with plus fertilizer was 38 pods. Now, the interesting part about that is we can have all the pods we want, but making sure we can fill those pods a lot of times comes up to moisture level, but also plant health and nutrition. But the interesting part about it, when we went to see this, we didn't look at yield, we looked at return on investment based off of each one of these. And you showed the control field, basically net income was $432 per acre. And you can see at this point on 728, it was already showing stress. It was cannibalizing itself, where the treateds were much greener, much less yellowing within the plants. But the net income of the T1 basically was $753 per acre. So even with the cost of utilizing the inoculums, we saw a higher return on investment. And the one with the most pods still showed an increase of $587 basically of return on investment per acre. It just didn't fill all the pods.
Yeah, we counted a few of those chickens before they had hatched.
Exactly.
Lots of pods they've got to fill.
But it also came down to nutrition.
It does.
Is we didn't have the adequate nutrition that was done early enough, which showed up on a plant sap analysis, which now you know for the next growing season.
A timely foliar in this situation would have helped fill even more of those.
Yeah. But we see this same thing. We're seeing this in our wheat trials that we're doing. And the cool part of this is we match the biomaker's DNA sequencing with plant sap analysis in the wheat field to look. And one of the things that we saw based off the biological plot index was phosphorus solubilization, inorganic P phosphorus solubilization increased by 15% over the control of more available phosphorus. And when you start to think about that, that's really significant, especially when you talk about all the environmental conditions that we have based off availability of phosphorus. And what we're seeing is this phosphorus is being made available earlier to the plant.
So we see the genetic capacity in the soil shift and change. That was 15 average. There were two different treatments in this case. One of them was up about 15. The other was up about double that. So the control is in biomakers' terminology, mentality, it's a medium. It's a normal level of phosphate solubilization genetic capacity. When we started doing one of the treatments, that jumped up to the high level and then up to the very high level of phosphate solubilization capacity genetically when we look at different treatments. And this is part of when we get into the idea of test don't guess. We've got to know what's going on with your plants, and that's part of why we target some of our approaches. The phosphate solubilizers, if you need more phosphorus, are going to do that job. If you need more nitrogen, we're going to use the nitrogen fixing bacteria, or sometimes a combination of the two. And the beauty is, as we upregulate some of that and we change the genetic capacity of that soil to allow more of that one nutrient, in this case we're talking about phosphorus, to go up, when we do that biologically and we do that through rhizophagy, we allow much more. But like we were talking about in the beginning, it's not just NPK. When that's our focus, it's not surprising that that's what the plant ends up getting. But we miss out on a lot of other things. So in the case where we reduced our application, about 60% reduction in nitrogen, significantly less nitrogen, 154 pounds versus, what was that reduced one at?
The one was 154, the one was 120, and we did 50, basically 50 pounds.
50, that's right, sorry. I should have taken better notes on this before talking about.
50 pounds.
Yep, 50 pounds. Okay. So we can see nitrogen up 17, but basically 18%, potassium 19%, magnesium 12%, calcium levels 20%. And that phosphorus up 14%. The calcium and phosphorus in this pH, it makes sense that they go up together. But we see N, P, Mg, Ca, manganese, cobalt, phosphorus, silicon, iodine, boron, molybdenum, selenium, all of those went up. And when we think about this from a biological standpoint of upregulation and allowing that plant to select the organism it needs, those organisms being a supplement, a 10-2-2 plus all of those traces, and through rhizophagy, that plant gobbling them up, farming those microbes, eating them, extracting nutrition, it makes perfect sense why we saw more balance and higher levels of all these other things, even though we're focused on N and P.
Well, the thing about it is, when we build our products, we have always said we build from a foundation with an emphasis on a given function. So in this case, if it's spectrum PSB, it's on phosphate solubilization, but it has all the other organisms there. And part of the reason for that is because they all support each other.
Yep.
Just like anything else, we don't want to go with the single microbe approach, the single nutrition approach. We want diversity and program out there. So we have those phosphate solubilizers, we have those mycorrhiza helper bacteria, we have those nitrogen fixers. We're getting all of those benefits with an emphasis on a specific function based off of data, based off of soil analysis and a plant sap analysis. So we have the phosphorus, we're not getting it in the plant. What do we need to do?
Yep.
This is what we need to do. This is how you build the programs to utilize biological function to enhance all the data that you have to help with overall soil health.
And then the rest of it is like gravy on your pie.
Yeah, you have gravy on your pie, Steve, or that milkshake.
Yeah, the plant milkshake material on top of the pie.
There you go.
All these nutrients is gravy. Oh man, we need to end.
Yes,
we need to be done. We're
Thank
getting
you,
pretty
everybody, for joining us for Natural Growing Through Biology. I'm Dennis.
I'm Steve.
And we look forward to seeing you soon.
Have a piece of pie. Bye, everybody.